In this session, I'm gonna overview protein engineering field. What is protein engineering? That is just to produce more valuable proteins. This field is a major sector of bio-industrial field. We must understand the role of proteins in life. Basically, proteins just execute all biological activities. In other words, any biological phenomenon just underlined by corresponding proteins. Sometimes it functions as building structure. As you know, the central dogma indicate role of, the relationship between protein and DNA. DNA just encode genetic informations, but those information must be converted into proteins for their actual activities by two processes, transcription and translation. All other important macromolecules like nucleic acid, carbohydrate, and lipids, they do actually carry out different functions in life, but their presence in life also determined by presence of corresponding responsible proteins. So what kind of features all organisms just share must be explained by the proteins. Everything, but we just look at some the types in this slide. The metabolism indicating all chemical reactions occurring in life. We want to see such aspect by looking at the enzyme in chapter 11. Secondly, all life forms, they have some response to the stimulus or environment changes. For this category, we're going to look at membrane protein and signal transductions in chapter 12 and 13. Thirdly, life, they just have tendency to maintain their life forms. So we're going to look at antibody in this category in chapters 15. Organisms, they proliferate, multiply their the species numbers. Some growth and development and genetics are related issues. We're going to see these things, some the DNA binding proteins and transcription factors in chapters 8, 9, and 10. Organisms, as you see here, they do have a very beautiful the shapes and colors, and they do have also very hierarchical the aspect of structural organizations made up of cells, further into tissues, organs, and individuals. So we're going to look at such structural aspects by learning the virus and fibrous proteins in chapters 14 and 16. Lastly, evolution also very important aspect of life. This could be probably dealt with throughout the courses. This slide just summarizes some major functions protein performs, enzymes, transport and storage of molecules, some movement, mechanical support, defense, signal transduction, and growth and differentiations. Proteins really carry out all the biological activities. But thing is, such functions actually are made possible by their unique three-dimensional structures. As you know, proteins, they just made based on the messenger RNA and the tRNA and that kind of stuff by ribosome. Newly synthesized proteins, they just fold into very unique three-dimensional structures. These final well-folded proteins, they just functionally active state. These two states basically does not show any functions, do not have any functions. So we must understand the three-dimensional structures of each individual proteins. All those information to make unique three-dimensional structures are actually encoded in amino acid sequence in this newly synthesized proteins, but the information to fold into correct structures are actually not well understood at this moment. Okay, kind of black box in this field. This could limit our capability to apply basic knowledge of proteins into application field. Given that we are very limited in understanding proteins, we might see how can we approach protein engineering. Okay, to get more valuable proteins, basically we do have two approaches. The bottom here, modify the pre-existing proteins, or we might do novel design new proteins. Okay, this field is very young still, kind of old history, but its importance is getting bigger and bigger in modern biotechnology field. As I told you, the basic knowledge about the protein structure and functions must be utilized to apply and to get better proteins. However, I told you we are very limited in such basic knowledges. In reality, in this field, you must understand the current status. Here, in this field, we still try to understand better the basic knowledges about protein structure and function relationships. And also, instead, we developed some practical techniques by which we could gather better proteins. Examples, for example, combinatorial evolution. In another aspect, also very important, all proteins must be experimentally determined for their structures, because we cannot predict yet. In this class, we're gonna learn basically three different types of knowledges. For initial 5 weeks, you want to learn about some general aspect of proteins, covering chapters 1 through 6. Starting week 6, we might learn each individual specific proteins, such as DNA binding protein, membrane proteins, signal transduction proteins, immune systems, and viruses. And also sometimes we might study some very important valuable issues. Those are determination of protein structures, NMR and X-ray, and secondly, trends for the protein structure prediction, engineering, and designs. Lastly, in this session, I'm going to just introduce some examples for the protein engineering. As you see, the green color represents the FC fragment of immunoglobulin G, and above, blue color represents protein A fragment, some fragment, which is capable of binding to green one. Okay, this one, protein A domain, has 3 helices. However, only 2, 1 and 2, can directly interact with green one, and helix 3 is not. Helix 3 just responsible to maintain correct conformational structures of helix N1 and 2. So without helix 3, helix 1 and 2 cannot be folded into correct three-dimensional structures, meaning it doesn't have any binding the activity to green one. So basically, in this scheme, the people just try to create protein A where only helix N1, 2 are present, and helix 3 is missing, but they want to just create the identical binding capacity. They just approach it 3 ways, starting with only helix N1, 2. Usually they change it in 4 positions, and next 5 different positions, and another different positions, and finally they've got the succeeded in making such proteins.